Gametogenesis is the process by which diploid precursor cells undergo meiotic division to become haploid gametes or sex cells. In males, this process is called spermatogenesis and produce spermatozoa or sperm. In females, this process is called oogenesis and produce ova or eggs. The process of gametogenesis occurs in the gonads and involves the following steps. First, multiple mitotic divisions and cell growth of precursor germ cells. Second, two meiotic divisions or meiosis 1 and 2 to produce haploid daughter cells. And third, differentiation of the haploid daughter cells to produce functional gametes.
Stage of gametogenesis. The process of gametogenesis occurs in 3 steps. They are as follows. First is mitotic division along with cell growth of precursor germ cell. Second is the occurrence of meiosis 1 and meiosis 2 for producing haploid daughter cells. Number 3 is differentiating haploid daughter cells for the production of functional gametes.
The primary spermatocytes is under the meiosis 1. The sperm produces cells in testes, grow and become primary spermatocytes. They are diploid cells in which they carry 46 chromosomes. This two meiotic division produces two haploid secondary spermatocytes, and these two haploid secondary spermatocytes is carrying 23 chromosomes. These two haploid secondary spermatocytes proceed to meiosis 2 and 4 haploid spermatids produce. The spermatids are transformed into spermatozoa or sperm by the process of spermiogenesis. At puberty, the primary oocytes meiosis 1 and forms the secondary oocytes in the first polar body. The first polar body is visibly smaller in size rather than the secondary oocytes. The secondary oocytes in meiosis 2 after fertilization and one large ovum in another polar body. At this stage, the polar body may decide to form two small polar bodies, and the three polar bodies only serve to collect chromosomes as waste. Only the larger cell or ovum can sustain life on fertilization.
Are the microscopic view of gametogenesis. First is the male gametogenesis. Seminiferous tubules are the site of spermatogenesis where germ cells develop into spermatozoa in close interaction with Sertoli cells. Seminiferous tubules of the testes are the starting point for the process, where spermatogonial stem cells adjacent to the inner tubule wall divided in centripetal direction, beginning at the walls and proceeding into the innermost part or lumen to produce immature sperm. The seminiferous tubules are surrounded by a basal lamina composed of extracellular matrix that serves to separate them from the interstitial compartment, provides structural integrity to the tubules, and regulates the function of cells in contact with it. Lady cells are the primary source of testosterone or androgens in males. Spermatogonia are a heterogeneous group of proliferative cells in testes that are precursor cells for mature sperm. Sertoli cells are the somatic cells of the testes that are essential for testes formation and spermatogenesis. Sertoli cells facilitate the progression of germ cells to spermatozoa via direct contact and by controlling the environment milieu within the seminiferous tubules. The main function of spermatocyte is to divide and produce immature sperm called spermatids. Spermatids control and regulate the functioning of the Sertoli cells. Sertoli cells control the cell differentiation, cell division, and metabolism of the germ cells, or the spermatogonia. Spermatids also control the cell secretions, from the Sertoli cells. Next is the microscopic view for the female gametogenesis or oogenesis. In females, most of gametogenesis occurs during embryonic development. Primordial germ cells migrate into the ovaries at week 4 of development and differentiate into oogonia. Oogonia enter meiosis 1 and undergo DNA replication to form primary oocytes. Theca interna. Theca interna cells are responsible for producing the C19 steroids that diffuse into the neighboring granulosa cells and serve as substrates for estrogen production. Granulosa cells play a key role in the female reproductive system. They help you get and stay pregnant. Granulosa cells produce reproductive hormones and support ovarian follicles. These follicles are small sacs of fluid in your ovaries that contain eggs. The follicular antrum. The follicular antrum is the portion of an ovarian follicle filled with follicular fluid. Appearance of the follicular antrum during follicular maturation is the first sign that a follicle has reached the next stage of maturation. It has changed from a primary follicle to a secondary follicle. Cumulus cells cover the oocyte and plays a vital role in fertilization. Oocyte grading is based on CC morphology and subsequent maturation is dependent on communication between CC with the oocyte. Zona pellucida supports communication between oocytes and follicle cells during oogenesis, protects oocytes, eggs, and embryos during development, and regulates interaction between ovulated eggs and free-swimming sperm during and following fertilization. Oocyte. An oocyte is a female germ cell involved in reproduction. In other words, it is an immature ovum or egg cell. It is one of the largest cells in the body and develops in the ovarian follicle, a specialized unit of the ovary during the process of oogenesis, folliculogenesis in the cortex.
Gametogenesis important event happenings in each stage. First stage, mitosis. In spermatogenesis and oogenesis, the primary germ cell reproduct. It provides new cell for growth. Mitosis event include chromosome condensation, formation of mitotic spindle. Division 1 of meiosis. It separates the pair of homologous chromosomes and reduce the diploid cell to haploid. Meiosis begins in prophase 1, the complex of DNA and protein known as chromatin condense to form chromosomes. Division 2 of meiosis. During meiosis 2, the sister chromatids within the two daughter cells separate, forming 4 new haploid gametes. This is responsible for the production of gametes, which are sex cells that are vital for process of reproduction.
Meiosis is a form of cell division that halves the number of chromosomes when forming specialized reproductive cells, such as gametes or spores. Meiosis involves two divisions of the nucleus, meiosis 1 and the meiosis 2. Stages of meiosis. Stage 1, prophase 1, chromosomes become visible. The nuclear envelope breaks down. Crossing over occurs. Stage 2, metaphase 1, pairs of homologous chromosomes move to the equator of the cell. Stage 3, anaphase 1, homologous chromosomes move to opposite poles of the cell. Stage 4, telophase 1, and cytokinesis, chromosomes gathered at the poles of the cell. The cytoplasm divides. Stage 5, prophase 2, a new spindle forms around the chromosomes. Stage 6, metaphase 2, chromosomes line up at the equator. Stage 7, anaphase 2, centromeres divide. Chromatids move to opposite poles of the cell. Telophase 2, a nuclear envelope forms around each set of chromosomes. The cytoplasm divides. The result of meiosis is the 4 haploid cells. Mitosis, it is the process of cell division during which one cell gives rise to two identical daughter cells. The process of mitosis is essential to life. It provides new cells for growth and replaces old and dying cells. Stages of mitosis. Stage 1, the prophase. Sister chromatids condense. Mitotic spindle begins to form. Centromeres move to opposite poles of the cell. Prometaphase. The nuclear membrane breaks down spindle fibers attached to the centromeres of the sister chromatids at the kinetochore. Metaphase. Spindle fibers align chromosomes along the equator. Anaphase. Sister chromatid separate. Spindle fibers pull chromosomes toward opposite poles. Chromosomes segregate at opposite pole. Telophase. Mitotic spindle breaks down. New nucleus forms around chromosomes at each pole. Cleavage furrow forms. Cytokinesis occurs, pinching the cell of two into new daughter cells.
Next, we proceed to comparison of mitosis and meiosis. Here is the Venn diagram shows the similarities and differences of mitosis and meiosis. Mitosis occurs in all organisms except viruses, while meiosis occurs only in plants, animals, and fungi. Mitosis creates all body or somatic cells like blood cells, while meiosis creates only sex cells or germ cells like sperm cells. Mitosis involves one cell division, while meiosis involves two successive cell divisions, the meiosis 1 and meiosis 2. Mitosis produces two diploid daughter cells, while meiosis produces four haploid daughter cells. Mitosis daughter cells are genetically identical, while meiosis daughter cells are genetically different. Mitosis creates a human cells with 46 chromosomes, while meiosis creates a human cells with 23 chromosomes. Next, we begin two similarities. Both are occurs in plants and animals. It starts with diploid parent cells. Both produce new cells and both cells undergo DNA replication. In the last, both have same basic step.